Good afternoon, everyone. I'm Dr. Emili Banerjee. Today, I will speak regarding recurrent implantation failure and genetic causes of it, genetic counseling, how it can help. And actually, I'll answer few questions asked by different patients here. So, the first question was, what is recurrent implantation failure? Is this same with IVF failure? To answer this, I will say that IVF reasons are there, and is recurrent implantation failure. What is it? When clinician transfer and is apposition, the next one is the attachment, and then invasion happen. When after transferring the embryo, it is not getting embedded within the endometrium, then we call it as recurrent implantation failure. In recurrent implantation failure, there are actually many crosstalk between the embryo and the endometrium. And most of the cases we consider that recurrent RIF, we call it in short, this RIF is happening because of the embryo quality. In around 70 to 80% cases, we consider because of the embryo quality, the RIF is happening, and rest around 20% is because of the endometrium reason. Okay, now I'm coming to the next question. The next question seen 1, 2, 3, 4, 1 to 6 grading is there. This is according to the stage of the embryo. The 1 is the morula when it is changing to the blastula, that stage, and 2 to 6 is different stages of blastocyst. So this is 1 to 6 grading. The next grading is regarding the inner cell mass from where our fetus is forming, and next grading is about the trophectoderm cell intactness. So A is very good, and then B is fair, and C is not that good. That is the grading system. So it actually explains whether the trophectoderm cell intact or not, whether the inner cell mass cells are intact or not, and in which stage your embryo is getting transferred. It is not telling anything regarding the chromosomal ploidy, whether there is a aneuploidy in any chromosome, whether there is a translocation in any chromosome. If it remain in the embryo, maybe in a higher mosaic stage, maybe it is present in all the cells of the embryo, so that can lead to implantation failure. Next, I'm going to the next question. Okay, so how you can test these? And if you, I told that regarding PGT, and PGT testing whether our embryos will be damaged. Okay, so we can check, we need to check that whether the embryos are chromosomally okay or not. That we can check through pre-implantation genetic testing. Pre-implantation genetic testing, it actually 3 types. For aneuploidy, we can check for single gene like we do in thalassemia, in cystic fibrosis. In all these cases, we do it PGT-M, pre-implantation genetic testing monogenic. And when we are finding that in recurrent implantation failure patient, there are some karyotyping abnormality like reciprocal translocation. This translocation thing or inversion thing, if it is there, then we can choose the embryo through pre-implantation genetic testing structural rearrangement, PGT-SR we call it. And it is not essential that in each and every case of IVF we need to go through PGT aneuploidy. Actually, regarding the guideline, what was just last year in 2023 European guideline, it was published. According to that, when mother's age we can proceed to, we can choose the embryo through PGT-A. Or if we find any karyotyping abnormality through in any of the parents in the couple, then we can go through PGT-A. It's not absolutely necessary. And in the question what the patient asked, that whether the embryo will be damaged at the time of PGT, actually in PGT-A aim is by laser we are taking the trophectoderm, the outer layer of the embryo, from there by laser, by the very efficient embryologists, the expert, they take this one 3 to 4 cells from the trophectoderm, and from there we are doing the testing in our genetic lab. And there is a minimal chance of 5% only that embryo damage can happen by the freeze thawing and all these things. In 95% cases, no damage is happening, and most of the cases the results are very good. So you don't need to be that much anxious regarding the embryo quality, as in 95% cases we are getting good results. Going to the next question. If chromosomally good embryo was transferred, then whether it ensure that my next IVF cycle will be successful? No, it is not ensuring. It only ensure to some extent, because in trophectoderm cell we are checking, so mosaicism may be there, but to some extent it can ensure that your embryo that is transferring, if we get that result is as euploid, that all the chromosomes are okay, then we can say that chance is there that the chromosomal aneuploidy will not be present in the fetus when it will develop in the prenatal stage. But there are some other reasons also there, that the microbiota of the uterus at the time of intercourse or in the IVF transfer, in the vagina, in the uterus, the microbiota also plays a very important role. Other infections and some other reasons are also there. So the crosstalk is very important between the embryo and the endometrium. If endometrium is also okay, is also helping in implantation, then by checking through PGT-A, we can get a good result. So going to the next question. How can I check uterine microbiota? Yeah, you can check the uterine microbiota before transferring the embryo. You can do that. From 2020 onwards, different research papers published in different good journals, where they are saying that the microbiota environment is very important. When the harmful bacteria are there, then it is actually producing some innovation to implant the embryo. Whereas the salmonella and other good bacteria are enriched, we are getting that, then we can see the implantation failure rate is very low. We can do this identification through real time. Whether the transfer time of the embryo have any role in successful implantation? Yeah, again a very good question. Yes, the transfer time is also there. Here we are coming to the actually endometrium part. When the transferring, actually we call it RIF, when there are 3 times of transfer already happened with good embryos, or 10 altogether 10 embryos transferred in multiple IVF cycle. When it failed, we call it as recurrent implantation failure. And it is happening, the embryos are good, we have checked through PGT-A, but again there are problems. So what we can do in that case, we can check the window of implantation. We call it as WOI. In around 20% of the people, 20 to 25% to be precise, in that percentage of people, we find that actually we transferred the embryo in 19 to 23 days within that. And what is the best time to transfer the embryo? We can detect it through ERA testing. We call it endometrium receptivity assay or endometrium receptivity array. Actually depends whether the company is using NGS technique or RACGH technique. It depends on that. First, it was started with checking only 61 genes, when these genes are actually differentially expressed. And now, if I say you the recent data, the differential expression of 616, if I am not wrong, 616 genes are being tested. And so, by differentially expression of these genes, we can measure what is the best time to transfer the embryo for that particular people. Actually, we need a healthy embryo, we need a healthy endometrium, we need a healthy environment also to make successful the recurrent implantation failure. From failure to make it a success, these things are very important, and other lifestyle factors are also there. You need to maintain the BMI properly. Alcoholism should be avoided, smoking should be avoided. You will get success, obviously. Thank you.